Morning and a very warm welcome to Aquafil. After an overwhelming response of our previous colloquium series, we are very happy to announce that this is going to be our fourth lecture series of this monthly colloquium, organized by Marine Ecology Laboratory in association with Aquafil, Presidency University, Kolkata. So I'd like to request Dr. Sumit Mondal to introduce our speaker. Over to you, sir.
Please present the introductory slide.
Yes.
Okay, good morning everyone. It is indeed my pleasure to welcome you all for this monthly colloquium series fourth lecture organized by Marine Ecology Laboratory in association with Aquafil. Today we have lucky to have with us Dr. Tushar Kaushik, Scientist C, Biodiversity and Paleobiology Group of Agharkar Research Institute, Pune. His research area encompasses various aspects of Foraminifera taxonomy, diversity, biostratigraphy, and paleoceanography. He has obtained his PhD in geology, micropaleontology specialization from Department of Geology, University of Delhi, and he has scored a third position in the NET JRF during his research scholar position and joined at ARI in 2015. He has many awards and achievements to his credit. Few of them are he was a participating scientist at Indian expedition to Southern Ocean 2020 and on board's SA Agulas from 10th January to 8th March 2020 at Indian sector of Southern Ocean and Fritz Bay, Antarctica. So I was also part of this expedition, and we had very good collaboration talks during that expedition period. He served as an invited subject expert in geology at National Institute of Educational Planning and Administration, Delhi, during 2018. He has participated and gave invited talk at 9th Regional Brainstorming Session for 36 Indian Geological Congress subject expert at molecular micropaleontology at NCPR Goa in 2017. He received Page's visiting travel grant for participating in 12th International Conference on Paleoceanography at Netherlands. He has executed 4 research projects in-house and extramural as a principal investigator and published 8 scientific papers in very highly reputed international journals. He has been serving as mentor for many research scholars for their projects. He was the organizer for the first summer school on paleontology, a window to ancient environment and life, in 2019 at ARI. He is a member of Cushman Foundation for Foraminifera Research and serve as ad hoc journal reviewer for Journal of Earth System Science, Springer. With this brief introduction, I am requesting Dr. Tushar to deliver his today's talk, an integrated approach for taxonomic reassessment in Foraminifera using morphological and molecular tools. I feel this talk will be very much motivational for the MSc and BSc students because he will be focusing on how morpho-taxonomy and molecular taxonomy can be coupled for a better research application. Over to you, Dr. Tushar.
Thank you, Dr. Sumit, for such a nice introduction. So I hope you will all like my presentation. So, am I audible, sir?
Yeah, you are audible.
So I'll start with the presentation. Can you see the screen?
Yes,
Yeah,
it's
it is.
visible.
Am I audible?
Yes, you are.
Okay. So let me start with a very good morning to all of you. Namaskar and hello. Let me start my presentation with introducing you all to my research institute, which is called as Maharashtra Association for Cultivation of Science, Agharkar Research Institute. So this institute was established in 1946 at as Maharashtra Association for Cultivation of Science. That was its parent name or the first name, and later on it was renamed on its founding member, that was Professor S.P. Agharkar, as Agharkar Research Institute in 1992. So going through a few, the mandate of the institute is to do multidisciplinary life science research. We are a community of highly experienced scientists and researchers of high repute working on genetic diversity of microbes, plants, animals, including fossils. So you can all visit us at www.aripune.org. So with that, let me start with my presentation. So, hello all, my name is Dr. Tushar Kaushik. As Dr. Sumit has already explained what I am and what I am doing. So, here I'll be just focusing on the presentation, and what I do is actually I explore new biodiversities of Foraminifera and implement the knowledge in understanding past or paleobiodiversity, or you can see fossils record also. So because the modern species provide an analog to the ancient or ancestral species which are now extinct, they are very useful to study modern analogs or modern living forams. So now let's begin our presentation, for which all of you have gathered on an online platform. I once again thank you and the organizer, Dr. Sumit, and his students for coming and inviting me. So the title of the presentation is An Integrated Approach for Taxonomic Reassessment in Foraminifera Using Morphological and Molecular Tools. So here is an outline of the lecture I'll be giving. So in this lecture, I'll be mainly focusing on Foraminifera, a model as organism which I really love and love working with, and has an immense potential in climate research, hydrocarbon exploration, as well as understanding past environment. I suppose this talk is attended by many students, as Dr. Sumit has already told in introduction. So I'll be giving a brief tour about Foraminifera so that they understand what they are and hopefully fall in love with them also. So what they do, where they live, how they are useful, and most importantly, why we all should study them. Secondly, I'll be dealing with research part associated with Foraminifera, showcasing gaps in current knowledge, problems with traditional morphological based taxonomic studies, finding ways to provide solutions to the taxonomic problems, and a new approach developed by our lab of combining morphological, which is traditional taxonomic ways, and the modern ways of molecular DNA based taxonomy, and the conclusions we have come up with and the progress ahead. The lecture will also provide an insight to the students about species concept in Foraminifera, problems related to cryptic species, a species which is morphologically similar but molecularly different, are called as cryptic species. Ecophenotypes means a species living in different environments but looks same. Problems with major taxonomic groups, lumping them as single species. Ways to understand the ways of evolution in Foraminifera, because they have a fantastic paleo records which goes back to 540 million years. The phylogeny and how we may solve them. Going over to the next slide. So what is Foraminifera? Foraminifera is a group of highly diverse single cell eukaryote. They are widely used as biomarkers of past and present changes in marine environment. Foraminifera are characterized by presence of small fibrous reticulopodia, which you can all see here on the left side corner photograph. And the test can be organic, agglutinated, or calcareous. Yes, they come in various sizes and shapes. They look scary and cute also. This is my perception. Once you start studying them, you will fall in love with these micro animals, also known as protists. So here is another. In the right side, you can see a video of Secominute. This is an organic walled foram. So basically, this is caught during our culturing experiment, and you can see on the very mouth of it, where you can see fibrous type things, it is feeding on a diatom. So the primary diet of Foraminifera is basically bacteria, but they also feed on diatoms, copepods, and other zooplanktons. So these small animals are cute as well as scary and as well as very, very deadly. On the left, you can see a Foraminifera attached to the red coral. And on below is that same foram which we have plucked from that red coral and is showing you in this. This is called Pararotalia. So going over to the next slide. So here I'll explain you where do Foraminifera live. So I hope by now all of you have started liking these small, fiercey, tiny animals. Like any enthusiast, you must be wondering, where do these forams live? Where can I find them? Well, solutions to your inquisitivity or query is that they are exclusively marine organisms. Well, this is another reason I am in love with these organisms. Because to catch them, to see them, to study them, my research takes me to the really fantastic, beautiful coastal realms of India or beaches, even in the open ocean, and I have even been able to lucky enough to visit Antarctica just in a search for forams, which I am sure you would love to see. Therefore, I will show you some of the images of our fieldwork at the end of the presentation. So carrying on with our presentation, Foraminifera are exclusively marine, but a few of them have been found to inhabit freshwater environments. They can be found from the coastal region of India, for example, a beach environment, to the abyssal region of the deep sea, as you can see from the image. Some of the forams are benthic, that means they need some sort of a substrate to attach themselves. I'll be explaining later why. Now, again, and some of them in this photo, as you can see on the right side, I don't know whether I have I can point that or not in this presentation. Not moving. Okay. So on the right side, you can see planktonics, which is my favorite organisms because they are the lazy ones. They are the passive dwellers. They don't have any cilia or flagella with which it can navigate and go left, right, forward, back. They just go with the flow of ocean currents. A good take-home message, that means go with the flow. Now, these organisms, as I said earlier, are important for paleoenvironmental research because they evolve around 540 million years, somewhere in the Cambrian age, when all living forms on Earth came into existence. It might be an alien, I really don't know. Also, out of 48,000 species recorded till 1992, 10,000 are reported to be still alive. So let me show you in detail what are benthic forams and what are planktic forams. So most of you might have visited a beach or a coastal region in India or and abroad. Now when you walk on beach sands, as you can see here, this is me, happy face. I love Foraminifera, that's why. And today I'm going to catch a few. So I'll be walking on a beach and also diving also to catch the forams. As I start walking on a beach, I encounter various species of benthic forams and a few of planktic forams as well. Now when I see my beach sand, which I have collected or grabbed through my hand under a microscope, I would see something like that. Image just popped up. Wow, a foraminifera in the center. Ain't that amazing? So now all of you must try next time whenever you go to a beach. What you really need is a dissecting microscope, sorry, to see a foram. And the beach sand, you must see what kind of a different organisms lives in the beach sand where you walk. And don't worry, they are not harmful. Once you take a shower, they will all wash away. So second is catching a planktic foram. So this is my favorite because it takes me to the research expeditions, which I really love to go. So catching a foraminifera requires a lot of tools and machinery, and it is a costly process involving boarding a research vessel. So here is a quote from a very famous movie. I'm sure you must have all have seen that. Big things have small beginnings, which is true for forams. So as you can see here in this slide, I am showing you that a ship is moving forward, and along with that, it is towing with it is called as a bongo net. So with this bongo net, we catch living forams which lives in the upper water column, that is around 100 meters or 200 meters. So we want to catch them live because we study live forams as well as dead. So now another way of catching them is through the multi-plankton net. So you can see on the second image which has popped up on your screen. This is called as a multi-plankton net. We used it during our Antarctic expedition. What it does is it goes up to the 2000, 3000 meters depth and open small nets, and then the nets can be towed back to the surface, and while towing back to the surface, it catches the live forams in that particular water column. For example, if we want to know what lives between 1000 meter depth and 1500 meters depth, we need this kind of an equipment. And if we want to know what lives in the upper water column, that is around 100 meters, 50 meters, we need bongo net. That will be more than sufficient. So what we do is, because we are catching them live, we are taking, suppose you take a fish out of a aquarium or a tank, it suddenly start dying up. Similarly happens to the forams. So we have to immediately process them, sit under the microscope in an ocean, in the research vessels, and start picking them individually, as you can see in this photograph. So these are the live forams which were caught during our Antarctic expedition. You can see they are small LED bulbs of different colors: yellow, pink, orange, blue, yellow, pink, orange, green. And that's why I just love them seeing under the microscope. And another way of catching forams, basically the dead forams, is through the drilling vessel, which can drill the ocean crust or the soft sediment in the ocean, sorry, which can drill this. So you can see, like, this is just an illustration how a drilling is happened. So the core goes down into some seamount or something where the sediments have been deposited. So sediments are mostly is a foram sand, like means 90% of the sediment will be foraminifera itself. So we are lucky enough that they are preserved in such a fashion. A lake care stratigraphy, one by one, age by age. So you just extract that core and start studying them if you want to see the diversity of forams in the past. For example, 1000 years, 10,000 years, 20,000 years, 50,000 years, and so on. So this is another way of sampling them. Mostly you will get maximum amount of planktonics in these kind of cores. And once you take out a core, you process the sample, the image just popped up, and you see under the microscope, you will see a foram something like this. So here is the, sorry. So here is the image of live forams, which look like bright LED lights. And here is the image of dead forams, which just look like white calcareous organisms. So it's really fun to study what a living one does and how they are feeding, what they are feeding on, what they are doing. And it's important to study what their ancestor was, to go back in time and analyze different kind of oceanographic thing, conditions changing and all these things. So it's really fascinating for us to study both live as well as dead. So now, as I've already told, Foraminifera are found worldwide only in the marine realms. So in this image, all that blue color are the places where you can find planktic forams in abundance as compared to benthic, and all the yellowish and the brownish ones are those regions where you will find benthic ones in abundance as compared to the planktic ones. So basically forams are found everywhere in the oceans, and the oceans are the only thing which connects every continent together. So it's really important to study the oceans. So here we have arrived at the most important question we must ask ourselves all the time. Why study? In this case, foraminifera. So if we want to understand ocean climate linkage, which is the biggest question now in IPCC, International Panel for Climate Change, and how marine ecosystems are going to change in future, which I'm sure Dr. Sumit might have already explained you in many of his talks. For that, we need to know what is the baseline variability, how the climate system and the ecosystem in the oceans are reacting to the change. And unfortunately, our historical records are simply too short. We need to look over time periods of hundreds, thousands, 10,000s, lakhs, crores of years. And for that, we need some kind of a record or a proxy of the conditions or events of the past. And this recorded in our case is Foraminifera. Foraminifera does have rewritten on them what has been the temperature of the ocean in the past, what was the pH of the ocean in the past, etc., etc. It has clues, and we all use this information as a proxy or a tool in an indirect ways as a proxy for past ocean and climate. Now, when I say proxy, it reminds me of very famous song from a movie Golmaal, not recent Golmaal, the old Golmaal. The wordings of the song is एक बार वक्त से लम्हा गिरा कहीं, एक बार वक्त से लम्हा गिरा कहीं, वहां दास्तां मिली लम्हा कहीं नहीं. So I hope that explains what I mean by proxy. Means something which was present at that time when some things was going on, and now we want to see back in the past what was going on. We need something really materialistic which has crystallized at that point and now stored as a geological record. So in that case, Foraminifera are the perfect organisms to study, because they have a fantastic geological record, exceptional preservation, allowing them to study evolution as well as extinctions, paleoceanographic studies, ocean climatic linkage, biostratigraphy, and hydrocarbon exploration. And for the living ones, which I have shown you in our past slides, like previous slides, species as shown in previous slides, we can study them alive in our laboratory, meaning we can know what they are doing, how they are making their shell, what do they eat, how do they live, how do they reproduce, what nutrients they prefer, are they dependent on oxygen as we are, or can survive in anoxic conditions also, because these questions are not provided very clearly by the dead ones. So we need to study them live. As I have already explained in previous slides, that modern organisms are an analog to study the past forams. So it's a combination between biological part and the geological part that we are doing here at ARI. It is remarkable if you look the size of these tiny animals, and they are less than a millimeter short. They are barely visible by a naked eye, yet the analysis only a few shell can provide clues to the global climate. So I am infusing interest into you students because it is necessary for us. It is our job to do that. So I'm again and again pressing on that aspect. So despite their large application of forams in geosciences, what we have found is that their taxonomy remained largely overlooked. Now, the paleo-environmental research in which Foraminifera is heavily used, which I've already explained as a primary proxy or tool, requires a strong taxonomic platform. So now for every research, let it be coral, let it be nematodes, let it be any other research in which an organism is the backbone or the baseline, the taxonomy is the fundamental thing. Suppose you want to work on some XYZ organisms, you want to work what they are doing, how they are behaving. For example, if you not know their proper taxonomy, your whole results can bring in lot of errors in your study. So taxonomy is the most important thing, and unfortunately not many people are interested in taxonomy, but someone has to hold the torch. So we at our lab are doing it, and thanks to our students who are enthusiastic enough to do the taxonomy. So let me give you a brief tour of the taxonomy of forams, how it has evolved. So the taxonomy of forams, like any other taxonomy, depends on some of the atlas, or we say as a bible of our research work. So here are a few of the examples of the taxonomic work, which taxonomic atlases which we usually refer. So now comes to the slide where I'll explain you briefly about the problems with the traditional morpho-taxonomy of Foraminifera. Now the till date, the foundation of all these proxies are based upon the preconceived assumption that each morphospecies, let it be anyone, for example in this case ammonia, it represents a genetically continuous species with a unique habitat and widespread biogeographical distributions. Means an ammonia found in Indian coast, an ammonia found in African coast, an ammonia found in American coast, Atlantic Ocean, Pacific Ocean, Indian Ocean are all same. A single species morphological looking are all same. The widespread general practices of lumping ecophenotypes means a same species, suppose A found in Indian Ocean, same species A found in Pacific Ocean, whereas both of these oceans have different salinity and temperature and different environmental conditions. They all are lumped into species A, and both are considered as different ecophenotypes, means environmentally different, or morphotype into a single species complex, completely ignoring the biology of the cell. So foram is the only organisms which has least studied in terms of molecular biology. I don't know why, maybe because the DNA studies have started very late in 1994 and so on. So in recent years, ribosomal DNA analysis has provided various evidences that the classical morphotaxonomic approach underestimates the biodiversity and ecological preference of planktic forams as well as benthic forams. These studies warrant a serious reconsideration for all of us, including the geologists, biologists, geochemists, for employing all species-specific paleoproxies for paleoceanographic and paleoclimatic interpretations, and may also contain a significant noise in their database. So this is a very important point. However, these molecular studies have mainly focused on the Atlantic Ocean, Pacific Ocean, Arctic Ocean, as well as Antarctic Ocean. And very few, mind you, very few investigations has been carried out in the Indian Ocean, including Arabian Sea and Bay of Bengal. Well, this might be due to the lack of technical expertise to carry out molecular studies in Foraminifera, or the study of paleoclimatic patterns with reference to molecular studies of Foraminifera might not be a primary concern in the research groups, but the things have started coming up. Our lab is the first lab in India which is using this kind of a combined molecular as well as morphological techniques to give solutions to many problems of the taxonomy, to provide a better background basis for further research. So this is a classic example. In this case, all the images you are seeing belongs to a genus called Ammonia. It was discovered by Bronick in 1772. It was the first genus assigned to Foraminifera and was originally described as Nautilus. So Nautilus is an ammonoid genus from the beach sand of Rimini, Italy, where these people were very enthusiastic to study what the sand look like, what organisms live in the sand. So the Cushman, 1926, was the first person who introduced the concept of ecophenotypy in Foraminifera. What he does was he lumped all those forams which look same from different regions of the world and make them as a species complex called Ammonia beccari, which has only 3 different morphotypes. For example, as Ammonia beccari forma beccari, Ammonia beccari forma parkinsonia, Ammonia beccari forma tepida. So this is just classic case. If you want to study more, you can search ammonia and its ecophenotypy online. You will get a lot of results. Another case is of the planktic forams. This is our study in 2016 by one of my colleagues, Srivastav et al. So what we have found in the dead samples, that is these white ones are the dead ones, as I have already explained to you, that the different taxonomic schools have employed different morphological criteria to define taxon, and or different formal names are adopted to define same taxon. In this case, Globigerina bulloides. The occurrence of various cryptic or polymorphic species or subspecies incorporating a range of gradational diagnostic morphological features, means similar looking features, inevitably leads to various different erroneous species identifications, leads lot of confusion. This in turn introduces error into foraminiferal based environmental reconstruction. In this case, Globigerina bulloides, all the 17 specimens you are seeing have been lumped together as one single species complex called as Globigerina bulloides. However, all the 17 specimens have been discovered, named, identified by different taxonomic groups as Globigerina falconensis, Globigerina umbilicata, Globigerina obesa, Globigerina quadrilatera, Globigerina megastoma, Globigerina bermudezi, Globigerina caryaconsis, and Globigerina riveri. So there is a huge disparity or huge problems associated with the identification of Foraminifera, which forms the backbone of all the climate-based research, all the paleo-environmental based research, all the paleoceanographic based research. Also, the problem lies with the understanding of evolution of Foraminifera. So what we do is we do ancestor descendant relationship by identifying conserved morphological features between the both in studying the geological time scale. Well, we have to test that, because if we have an additional technique called as molecular technique, we must test that whether it is true or false. There is no harm on retesting. That's why it is called as research, because one has to find. So in our lab here, we are trying to give solutions to the taxonomic problems. That's our scientific job. What we do is we integrate foraminiferal taxonomic framework by integrating, by combining, as you have seen, plus marks, by combining traditional morpho-taxonomy, which has been practiced for so many years. We are not ignoring that. We are incorporating into it, and we are adding additional information of molecular systematics using DNA of living forams, and erecting a robust taxonomy of a particular species, which is a very tedious process, I must tell you. So here is a results of few of the published literature from various authors world over. So the work is not new. It has been, as I've already explained, it is being done and practiced in different parts of the ocean of Pacific, Atlantic, whereas Indian Ocean is very less studied so far, as per my best understanding. So these are some of the examples. Here on the left, you can see a species called Globigerinoides ruber, which has different morphological shapes. And on the right, you can see it's called Globigerina bouloides. So people have found out 4 genotypic types in Globigerina bouloides so far. There are 3 to 4 genotypic types in Orbolina universa, which is just a ball having different pores in it in which forams exchange food from the Caribbean Sea, Mediterranean Sea, Sargasso Sea. So the take-home message from this slide is your data correct? As I've written and bolding mark in red. So one has to clearly incorporate these kind of findings into their research if we are doing geochemical research, paleoceanographic research, because taxonomy is the most fundamental part. I'm again pressing over it, stressing over it, because it's the most important, which is being not practiced much. And if you don't practice it, then there will be an error in your isotopic or elemental analysis for reconstruction of paleoceanographic or paleoclimatic results. Hence, these studies warrants a serious reconsideration. So one must always consider taxonomy as their prime focus before starting this geochemical or paleoclimatic research. So recent developments in taxonomy of Foraminifera integrated morphological and molecular approaches. You can see on the left is a traditional way of classifying Foraminifera. So this is the traditional classification by Lovelace and Tipton 1992, which I am sure all the Foraminifera people who works with forams are aware of it. This is our Bible. This is our everything if we want to study Foraminifera. But what has come in the future is people have extracted DNA from different species, different genera, different orders, and different classes of forams, as well as other eukaryotes and protists as well, and they have come up with a totally different classification in which they have classified all the forams into 3 orders, which is monothelemids, Turbothelmia, and Globothelmia. Whereas in the original traditional taxonomy, they are classified based on organic test, agglutinated test, calcitic test, and in calcitic test, they are further classified as triserial, biserial, macroperforate, microperforate, and further classified as non-spiral, non-spiral, non-spinos, spinos, trochospiral, perispiral, etc., etc. Basically following the shape or the composition of the test. So now the beauty of doing molecular as well as morphological integrated taxonomy has resulted in exponential rise in number of taxa discovered since the advent of this kind of techniques. As you can see, since 2004, 2005, the peak is almost flat. Not much work is being done in this aspect, but and henceforth there you can see an exponential rise in discovery of taxa, accepted taxa, distribution records, literature sources, and the images available on the online platforms. So the take-home message from the slide is that discovery of a new genus or species of Foraminifera bring with it new opportunities to improve the biostratigraphy as well as paleo-environmental reconstructions. So now what we do at ARI are just simple a flow chart of what really we do. I'm not going much in detail. My basic motive was to infuse interest in students and researchers to undertake taxonomy as their primary objective. So this is a photo of our lab. Here is my student working on a microscope seeing forams in it. And on the right, you can see a flow chart. So what we exactly do is we collect forams, live forams from various beautiful locations all along the coastal Maharashtra and also around the east coast of India in the Sundarbans through collaborations. Then we study them under the microscope. We try to identify living versus dead forams. So here is my student looking under the microscope and observing the forams. And then what we do is we take their SEM photographs to erect a robust taxonomy to see more clearly, more detailed characteristics, morphological characteristics of the species. Here in this case, I am showing you Quinquiloculina seminula, which is a benthic porcelaneous foraminifera. Then what we do is we do their DNA extraction by following different buffers, which is DIC or either DOC or urea. and then we extract them using polymerase chain reaction or PCR machines. Already know, I hope so. And then what we get is a DNA sequence in form of a chromatogram and converted into this GATC format. And then what we do is we combine our traditional morphological understanding of a species with integrated it with the molecular results, and then we come up with our results. So this is the final slides of our presentation. Here I'm showing you, as I've already explained several times, the beautiful places which takes me in search of forams. So taxonomic research is exciting and fun as well. It's not just boring sitting into books and looking and peeping into microscopes. So what you really get a chance is to go and visit various different places all around the world, and you can catch them, you can catch the forams, you can enjoy the views. So the take home message is the search for Foraminifera takes you on a mesmerizing adventure which stays for a lifetime. So in this image, if I may explain you, that on the right you can see images from our recent Antarctic expedition, in which me and Dr. Sumit and my student here, Mr. Mangesh, have participated. On the left you can see my previous student, Mr. Thirumalai, is doing fieldwork along the coast of Maharashtra, and here is me doing scuba diving, collecting benthic forams from the depth of 10-20 meters. So taxonomic research is exciting, fun, and rewarding as well. So we have been lucky enough to get several papers out of our research, and we are the first Indian group to contribute more than 60 DNA sequences in a GenBank database, in which every sequence all around the world of foram or any other organism is submitted before it is published. So thank you everyone for listening to this presentation. The final take home message is integrated taxonomy is equal to taxonomy. And you can contact me on my email ID shared on this presentation last slide. If you have any further doubts, I am happy to take your questions. Thank you.
Thank you, Tushar, for a nice and innovative talk. I want to say one thing that to all the students, that Tushar is the example of how the subject doesn't matter, and nowadays the integrated approach is everywhere in the science. He is basically geologist. Now you can see from his presentation, he has moved towards the biology part. So those students who complain that my subject is this, I don't want to shift to another subject. So for them, he is a classic example. So being a geologist also, he is doing wonderful research on molecular taxonomy, so which is entirely different ballgame from initial days for him. So however, he has succeeded in this, and now he is publishing very good, nice papers. And from India, he is the only pioneer in this doing molecular taxonomy for Foraminifera. So thank you, Tushar, for pointing out that taxonomy is not a dying science, and students will be, I think, will be motivated to take up the traditional taxonomy in coupled with the high-tech or modern molecular approach. So I have one personal question to Tushar, that what is your opinion that in these forums, how it will be helpful for finding that, suppose in the land, any land portion where these areas were under the sea or previously, how do these Paleoceanographic study contribute in these foraminiferous applications?
Yes, thank you for your question. So I repeat your question. Basically, you want to know if any area in the ocean, can we say whether it was earlier submerged or it was exposed to the surface?
Yeah, that is the question.
Okay, so there is a technique called benthic versus planktic ratio. So when we walk on the sand and along the beach, what we get is more of a benthic forams and less of a planktic forams. But we as go towards further into the ocean, into the deep ocean, the things completely reverse itself. We get more planktic and less benthic. So this gives us an indication or can provide us the mathematical way of finding out whether you have more planktic or less benthic. So at suppose any place, suppose X, If that place was earlier submerged, you will get more planktic as compared to benthic. And suppose after some sort of a tectonic activity or something, that place might have been abducting onto the surface. So eventually you will start getting less planktic and more of a benthic forams onto it. So this is how, based on planktic benthic ratio, you can really calculate that whether that place was earlier submerged or abducted onto the surface.
Moreover, I think that magnesium calcium ratio also contributes significantly in that.
Yeah, that is a secondary approach, but basic primary approach, which was fundamentally taught to us, and we know that is the benthic planktic ratio is the much easier way of finding out. And later you have many sophisticated techniques to find out these things. So it is endless.
Okay, thank you, Tushar. So now I will over to Momita for taking up the question answer session.
Thank you, Dr. Kaushik, for such an informative talk. I must mention our deep sense of appreciation for giving an excellent coverage to the topic entitled An Integrated Approach for Taxonomic Reassessment in Foraminifera using Morphological and Molecular Tools. It was so fascinating how Dr. Kaushik enlightened us on this topic and made us aware of so many interesting and unknown facts regarding Foraminifera taxonomy, diversity, and their evolution. So now it's time to move on to the question answer session. So first question is from Divisha Bishash. She has asked that are forams found in the Mariana Trench? So how do they survive in that pressure?
Okay, so thank you for your questions, first of all. So Devisha Bishwa, I would like to inform you that I'm really sad to inform you that forams are not found in Mariana Trench. So there is a level called carbonate compensation depth, which is also known as CCD in the oceans, which varies from place to place. And below that, you won't get much of the calcitic forams. However, in the Mariana Trench, you get radiolaria chert, which is also known as the red chert, which is another form of microfossils, and they are abundant in Mariana Trenches.
Okay. So next question is from Bhaskar. Why benthic forams like ammonia sp sometimes found to be in the surface water? What are the possible reasons for this?
Well, I'm not sure what he meant, but the benthic forams are basically found along the coast of any continent, and the ammonia is the most commonly encountered benthic foraminifera. So, what I would like Bhaskar to just go to any beach near his place and collect a handful of sediments and see under a dissecting microscope. I'm sure he will get some of the ammonia specimens in his samples.
Okay. So last question from Shomalo Chakraborty, that what kind of sequences are being targeted for identification of the species?
Okay,
May we
very
know?
interesting and I'm thankful for asking this question. So what we are targeting is the hyper variable regions in the DNA, because if we want to suppose, if we want to know that who was the ancestor and who was the descendant and how two species can be separated, we need some sort of a variable region on which basis we can dissect or bifurcate two species. So these regions are called as 18S, which is smaller subunit regions. Also, there is a region of larger subunit in the eukaryotic DNA, which are basically targeted for our research.
Okay, so this is the end of the question answer session. Furthermore, this will be available on our YouTube channel for further viewing. On behalf of Marine Ecology Laboratory and Aquafield, I would like to express my gratitude to our eminent speaker, Dr. Tushar Kaushik, for giving his valuable time and sharing his thoughts and experiences. We are so honored. Finally, I extend a big thanks to all the members of Marine Ecology Laboratory for their continuous effort and hard work to make the session successful. Thanks to all the registered participants for their cooperation and support. So lastly, I would like to request the participants to stay tuned at our website for upcoming events of this ongoing colloquium series. Thank you very much.
Thank you. Thank you so much.